Thank you very much, Gonzalo, for the invitation and also presentation. So today I'm going to talk about a work I did back in Argentina with Diego and Ignacio from the University of Buenos Aires, and also Carlos Bueno and Peter Wellness from Rice University. And it's about probing the dark energy in the functional protein universe. So, if I can move my slides, that would be nice. Yeah, here I am. So there's a recurrent flow of information in proteins. I want to start with this picture. So you know that protein sequences, amino acid chains in the proper environment can fall into stable, robust 3D structures that we can beautifully picture as the one I'm showing in the screen. But as you may also know, these kind of frozen pictures can actually work. They need to be dynamic, even locally unstable, in order to perform very particular chemical activities, and in a particular environment also let the organisms to perform biological functions. Throughout years and years and evolution, all these maybe conflicting features like forming a stable structure, but also allowing these local instabilities are being coded into the sequences because they're like constraining the actual protein sequences we can find up to date in nature. So let me show you maybe a window into this flow of information. This is a deep mutational scanning. So what is this? This is an experiment by Rocktin's lab from 3 years ago. It's a mega scale experiment, so they did this for many proteins. And in particular for this short protein domain I'm showing you here, they have mutated the protein to all the possible alternatives in all possible sequence position, and they are computing the following stability changes, delta delta G, simply. So for instance, in position 10, if you mutate the wild type leucine to a proline, you will have a destabilization of more or less 2 kilocalories per mole. And you can do this for all possible mutations, and you can see these interesting patterns with some particular positions that if you mutate them, for instance, this one, you will get mostly a destabilization of the protein. And you can also paint the structure with the weighted average of this same signal. You will see that the protein core is highlighted in red, which is expected for protein folding typically. But we also can take a different view, a different cut of this flow, which is, let's say, synthetic deep mutational scanning. This is the one below is completely synthetic, evolutionary, sequence-based. These are like the ESM2 zero-shot scores. ESM2 is a protein language model. And what we are seeing here is the logits, the wild-type logits, sorry, the masked logits of the model for every particular sequence position, and for mutating them to every possible amino acid. So in the same position, I was telling you before, position 10, if you mutate the leucine to the proline, you will have an increase in deleteriousness of more or less 3 arbitrary units. So the new sequence, the mutated sequence is less likely to exist for this particular fold than the original one, according to this model. And you can also paint the structure in the same way, and we can see actually a similar pattern between this experimental folding stability experiment and our sequence-based model. So one possible question is, are these two things the same? Are evolutionary scores and folding stability changes the same thing? Well, Pam may say yes. Maybe we need some theoretical insights for understanding the answer to this question. So first of all, maybe a nice framework for understanding this question is the energy landscape theory of protein folding. Here on the left, I'm showing you the energy landscape for a random heteropolymer. So this is not a real protein, just a random chain of amino acids. And what's expected for this kind of system is a frustrated landscape. So here, if you measure the effective energy of the system, you will find a lot of minima. It's a frustrated landscape because you can have like very slow transitions between all of these misfolded states that this chain is going to acquire. This can be characterized as a glass transition. So in this case, the glass transition temperature of the system is above the folding temperature. So the glass temperature, transition temperature is above the the folding temperature, you will find a behavior like this. But if you consider then evolution, natural proteins present completely different landscapes from this. And these kind of funnel-like landscapes, like the green one here on the right side of the slide, are typical of natural proteins. And in this case, you have still some roughness, but you can also you should also have a large enough energy gap. So between the native protein state and the random global state, you have this large enough energy difference. And in this case, you are folding the protein before entering into this glass transition. So you have some remaining roughness, but the energy gap is large enough. This is known as the minimal frustration principle. So what will be delta G, what we were measuring in the DMS in this context? Well, there will be like perturbations to this native protein structure. We'll have like a slightly different landscape for these mutated versions of the protein with different levels of E-fold. So we can approximate that the differences in E-fold will be the delta G you can measure in the lab. Well, what about the evolutionary model? Maybe we can add an interpretation in terms of landscapes also for these kind of models. So for a protein family, if we assume that sequences are in equilibrium, so this will be the case, for instance, for a multiple sequence alignment, then the probability that this kind of models is learning is given by a Wolfram distribution. And the Wolfram weight of the distribution is known as a free entropy, in this case, or simply an evolutionary potential, let me say. We are naming this psi evo. So psi evo will be the evolutionary potential of a particular sequence. If we train a protein language model, we cannot then access simply the evolutionary potential of a sequence, but instead, what is interesting is that we can access the difference in this evolutionary potential between a wild type and a mutated version of the protein. So if you mutate from amino acid A to amino acid B in position I, you can compute the mask logits, computing the probability masking this position for the new amino acid, the old one, take the log of the ratio, and you will get something like this difference. So if we plot right now, instead of the folding energy we were having before for the folding landscape, and an evolutionary potential, we will find this evolutionary landscape. And what we are measuring with the PLM is approximately this difference between the energy state of the natural sequence and the energy state, maybe a bit higher, of the mutated sequence. Above everything, we will have the random sequences characterized by a zero evolutionary potential in this case. So this landscape is the same for a single fold, and we have like different sequences in different places. Before, we were having different structures in different places for the same sequence. So can we combine these two approaches to the problem? Well, if we can assume that folding was the only constraint on evolution, and this is pretty reasonable if we consider the minimal frustration principle, well, given the high dimensionality of the problem, the sequence space, the probability of that a sequence falls successfully to a given fold, so for a protein family, is also Boltzmann-like. This is easy to prove, but so if you follow me, you will have you can write the probability of finding a sequence that is folding successfully in this way, and the Boltzmann weight here is given by E-fold. E-fold, remember, was the energy gap between the molten global state and the native state. And the scale is given by this KB T-cell. T-cell is a selection temperature, we will say. So if you do this, then simply our evolutionary potential is a rescale version of the folding energy. The last names are comparable, and then you can compute, and people have done that, the selection temperature for different protein families. And we know that the selection temperatures is linked to this glass transition temperature, and folding temperature should be lower than that, than those, for ensuring that evolution can find sequences that can fold. So then if folding is the only constraint, we can compare these two things, and actually these two DMS should be really similar. However, it's interesting that folding is not always sufficient to perform all the biological functions. We know binding, for instance, catalytic activity, the need to avoid aggregation, or allosteric moves in proteins. All of these are like functions that need more than keeping the protein fold. So in order to address this consideration, we need to define a new concept, and it will be the dark energy. Dark energy will be the difference between simply the folding and the evolutionary energy landscapes. So in this case, I'm drawing the landscapes again, and I'm showing you that there is here maybe a difference I'm characterizing as a dark energy between the native state in the evolutionary landscape and the native state in the folding landscape. The scales are matched because I'm considering the folding selection temperature. Maybe this is easier to understand if we consider variance, because if we mutate the protein, we will have a change in both of these energies. So we'll have a different folding landscape, and maybe here we are having a deleterious variant because we have a higher energy level in the purple landscape, the evolutionary one, and this is deleterious and also is destabilizing the protein because the energy level of the green one is also higher, and it's all the deleteriousness, all the sequence unlikely that we are provoking with the variant is given by folding. The dark energy will be preserved by the variant. But if instead we have a variant affecting, for instance, the active site of an enzyme, we can expect that maybe the destabilization is not that much, but the new protein with the new amino acid is less likely than the previous one. So this one has a much higher purple energy state in the evolutionary landscape, and the difference is larger. So we have a larger gap between the two of them. So then the difference in dark energy is explaining this variation in the function that is beyond the folding constraints. And we have to, if we go back to the first slide, we can understand that dark energy change for single site variants is only the difference between these two DMS I was showing to you before. So first of all, here is the ESM2 evolutionary DMS, the mass logis, and here the delta G. So to compute dark energy, we need to rescale the first one using the folding selection temperature. Folding selection temperature is being computed as the ratio of the standard deviations of these two distributions. And then once you have that, it's only subtracting. So this DMS rescale minus this one is going to give you the DMS of dark energy. So now the dark energy change is also showing us patterns, but in this case, patterns that are related to the function beyond folding. And also the scale of this new energy, dark energy, is also kilocalories per mole by definition. So now we have not only a score, but an energy measure we can apply to any protein for which we have delta G and the logs of an evolutionary model. And you can see that if you compare these two patterns again, you will find that maybe in the core of the protein they are similar, but maybe in this part they are not. And this is why dark energy is high in this region. So then we can start to measure this missing energy in the functional protein universe. So we are leveraging here Rocklin's lab mega scale dataset, the measurements of delta G for all possible variants of a couple of hundreds of proteins. And here I'm showing you some examples. You can see that dark energy patterns are different protein to protein. Sometimes you have highlighted in red helixes, beta strands, other times, in other cases, loops. And also, we can define a threshold for identifying which variants or which sites are having significant dark energy. And we found that 25% of the variants are presenting high dark energy in this data set. However, we have to know that this functional protein universe is maybe a bit reduced because we are like relying on wet lab measurements for delta G. What we can do is to use instead supplementary a model for computing E-fold, so the folding energy gap and the difference of the folding energy gap between wild type and mutant for any other protein. We're using here Awesome. Awesome is a very simple coarse-grained force field that, so I'm not going into details here, but it's transferable. It's pretty cheap to run. And it also has been trained, the matrices building also has been trained to maximize the ratio T-fold over T-glass, which is essentially to ensure that the folding landscape of the protein is funnel enough. So this is essentially what we need to ensure proper folding in this theory. So if we compare the results for the protein we were studying, patterns essentially match, and we have a decent correlation between dark energy computing one way or the other one. So let's use OSOM to explore dark energy in a more maybe interesting system than these little domains with experimental measurements. An interesting system in particular are enzymes. Here I'm showing you TEM1 beta-lactamase. Here the colors are, again, the weighted average of dark energy for this protein. These yellow balls are highlighting the active sites of the enzymes. As you can see, everything around the active sites is pretty red, and that color is like smoothly transitioning to white and light blue as you are like going away from the active sites. In this map, I'm showing you essentially the same values. I'm highlighting in yellow, again, the active sites. It's also interesting that there are also like other regions, maybe in light red, and that may be indicating like the presence of other functional constraints besides the active site. We run this on the catalytic site atlas. This is a database where enzymes are annotated according to their active sites. And we found that in general, in this database statistically, high dark energy changes are really enriched around active sites, and up to 88% of the variance in the active sites present high dark energy. And this number is going down smoothly as you go out in shells from the active sites. But it's also notable that in the outer shells, we still have 39% of the variance presenting significance and dark energy. So other things may be happening to these proteins besides the function given by the active sites. So what else? Well, I talked to you before about the binding selection, the folding selection temperature. We can compute now a binding selection temperature. What is the difference? So the folding selection temperature was measuring the relevance of the folding constraints during evolution. Now we can do the same with a function, in this case binding, but we need the function to comply with some constraints. So first of all, we need an experimental number for the functional energy. We need that to be possible to be measured and actually to have been measured in the wet lab. And in that case, we can write down the sequence probability distribution in a more, let's say, accurate way than the one we had before. Here we have a term for folding and a term for function. And we have the folding selection temperature and also a functional selection temperature that we can compute easily in this way if we have the dark energy. So for doing this, we are applying the theory to a very simple system, the bar-star-Barnard system, because we know that bar-star is essentially constrained during evolution for binding Barnards, folding, and not many other things. And we also have first lab data. These are very old papers, but very accurate. And if we compare the binding delta G they have measured in the wet lab to our dark energy changes, there's a very strong correlation. And from this correlation, we can obtain the binding selection temperature. We did the same for several binding pairs of protein from the SCEMP database. Here I'm showing you some examples, the ones where this correlation is strong, meaning that the functional constraints are concentrating in this binding activity and not other things. And what we found is that binding selection temperature is also in the same range as the folding selection temperature, which is actually expected because physicochemically, the activity of binding is not very different from folding. So we can imagine that the selection temperature for this function should share the range with folding. So finally, to conclude, let me wrap up. So we have defined a dark energy to quantify the functional constraints beyond protein folding, and we have that in a meaningful physical scale. in kilocalories per mole indeed. Also, we have seen that dark energy is particularly high around functional sites, for instance, in enzymes, active sites, in binding interfaces, as the one I showed you before. And also, functional, in this case, binding selected temperature can be defined and measured in simple cases and compare with all the other physical and effective temperatures. As a future directions, we may now imagine that maybe we can apply this same framework to predict or to disentangle maybe the molecular mechanism involved in every single subvariant, at least to distinguish stability-related variants from solely functional related variants. So I wanted to thank you all for listening, but also to my collaborators in this work, Ignacio Sanchez, Diego Ferreiro from the Protein Physiology Lab in Buenos Aires, Peter Wilnes and Carlos Bueno from Rice University, and also my current group, because I'm working in the Diaz and Fraser Lab at CRG in Barcelona. It's our probabilistic machine learning and genomic groups. So we are also trying to discuss all the things related to genomics right now. So that will be all. Thank you.
Thank you, Ezequiel. Nice presentation. So we have one question already. Other people who are interested, please write them or raise your hands. So Chandan is asking if can we predict the molecular mechanism of electron transfer from this? Like the transfer of electron from NADPH to different substrates?
Well, so the thing is, we can absolutely distinguish between the folding mechanism as a constraint and everything else. Then to deconvolve between several functions, maybe not that simple. If you are talking about a system where we have a single clear function affecting evolution besides folding, it's possible, and then we can try to rescale and compute something like the selection temperature we are measuring now if we have a physicochemical energy. If we have like several things happening at the same time, constraining the same protein sites or the same interaction network, maybe that may be a bit different. So then the dark energy will remain dark partially in that case, maybe.
I have one question in the meantime. So you mentioned that 88% of the catalytic residues, as annotated, I don't know how, show high dark energy. But then that gives you 12% of residues in the catalytic sites that do not show high dark energy. So I don't know if this is random energy or around zero or really low dark energy. So my question is, have you checked if is there any role in the catalytic active site of these residues that makes them as low energy? Is this maybe a misannotation of the catalytic sites? Maybe these are not really part of the catalytic site. Do you have any further insights into this?
Well, thank you, Gonzalo. It's a really interesting question. We didn't look at those sites in particular. I have to say, however, that these are fractions of variants, not sites. So maybe we have like different variants in different sites, but maybe it is the case that some annotated residues in particular are concentrating these variants. That is completely possible. I should say also that the general idea that we have on catalytic sites where they are like constrained for function and not for folding, and maybe not only non-collaborating with folding, but sometimes going against it. It's a general feature, but it can be a case where you have an active site which is maybe annotated correctly, but it's also contributing to fold. Then when you mutate it, you are affecting both folding and also the catalytic activity on the enzyme directly, if you know what I mean. So that is also possible. All these three cases may be interesting to look at, and that's absolutely something we can do.
Do we have more questions from the audience? You can also raise your hand if you want to use a microphone. Just wait a bit because sometimes people are shy. All right. We will have Ezequiel's talk uploaded to YouTube, I think. So you can also watch it there. And we thank Ezequiel again for such a nice talk. I think it's very, very vanguard. So thank you, Ezequiel. And now
Thank you.
we are going to, unless you want to say some words, I mean, we have extra minutes if you want to say something extra about your presentation. If not,
no, yeah, maybe that there are like a bunch of papers in recent years trying to explore the same classic question with different approaches. I hope that ours, which is maybe a bit more theoretical in some terms, is helping to find some consensus and common ground of all these analysis that these new data and models are allowing us to perform.